Hello and welcome to this short teaching ideas video on how to teach translocation in plants to A-level biologists. Right, some key things that you need to think about before doing any of this is to make sure that you've got all the bits made up and that they're nice and big. Right, there are many different cells that you'll need to make up, but luckily most of the cells or most of the plant cells that you've made up for this are interchangeable with other model ideas. Make sure that you're using tiddlywinks to help represent different ions that are involved, and it's worth cutting out some hexagons to help represent the glucose and the sucrose that's moving around. Finally, because this stage takes about 17 parts or 17 different stages within it, make sure that you've got lots of small broken down annotation cards. After you've done this, it's worth letting students photograph what you've done and getting them to go and produce their own diagrams from this, which they can then go and help them explain the process to another partner. Right, hopefully the next short clip will help show you how this type of modelling activity can be used in practice. Right, so translocation is the movement of sucrose through the phloem sieve element from a source to a sink. We start off, right, so the first thing when you're answering a question that you need to talk about. So the phloem sieve elements have gaps at both ends called plasmastomata. The plasmastomata allow the movement of sucrose and minerals from one sieve element to another. Right, the problem is though, phloem sieve elements, they're basically hollow cells. They don't have any organelles of their own. So they need companion cells which provide everything they need. The companion cells contain lots of mitochondria because they require lots of ATP to be formed because they're going to be carrying out lots of active transport. So the ATP is used to provide energy for a hydrogen pump to go and actively pump hydrogen out of our companion cells. So the hydrogen pump is going to be actively transported out due to lots of ATP being produced by the mitochondria. So this causes concentration gradient. So the concentration gradient of the hydrogen will be higher than the concentration inside. This causes the hydrogen ions to move back into the companion cells through a channel protein. As it moves back through this channel protein, it goes and it carries the sucrose into the companion cells from the surrounding areas. So the concentration of sucrose in the companion cells is increasing. Right, we can also call this, because it's moving back in, we can also call this facilitated diffusion, because it's being facilitated by a channel protein. So the sucrose concentration inside the companion cells increases. Because you're increasing the amount of sucrose in there, that's increasing the amount of fuel that's basically there for the mitochondria to carry out respiration. So this enables the mitochondria to go and produce more ATP, so it carries on pumping out more hydrogen ions, which can then go and move back in and so carry in more sucrose. So the more sucrose you have in the external area, the more ATP you'll end up producing, so the more sucrose can be transported in. So the sucrose concentration is higher in the companion cells than in the phloem sieve elements. So therefore the sucrose will go and it will move into your phloem sieve elements through the plasmastomata. So this increases the concentration of sucrose in our phloem sieve elements. So as the sucrose moves into the sieve element, this causes the water concentration in the sieve element to become lower. So therefore, your water concentration now in your phloem sieve element is going to be considerably lower than it will be in the xylem. So water will go and diffuse out of the xylem into your phloem sieve element because it will move down a concentration gradient. So this large volume of water that moves into it causes hydrostatic pressure. This increase in hydrostatic pressure enables the sucrose to be dissolved in the water, and because your hydrostatic pressure is always going to push things from an area of high hydrostatic pressure to an area of low hydrostatic pressure, this will start to move the sucrose from this area where it's a source, so where it's being made, down the plant until it gets to a sink, so where it's being stored. So once the hydrostatic pressure decreases at the sink, the sucrose, so the water, will go and diffuse out of the phloem sieve element into the sink, so somewhere in a route. That will carry the sucrose with it, and the sucrose will be stored in those sink areas. Hopefully this video has given you some ideas how to teach this particular concept. If you've liked this video, please subscribe, like the video. There's links below to follow me on Facebook, Twitter, and there's a link below to my shop on test where you can go and get a full copy of this lesson. Please feel free to leave any comments or questions below and I will get back to you as quickly as possible. Thanks for watching and happy teaching.